So we are now going to speak about the acquired immunity. So I alluded to the fact that there is a collaboration between the adaptive and the acquired immunity. We also have to have an understanding that these things are more dynamic in the sense that they may happen almost at the same time. But we can also create a sequence. For example, we can use that image of the local inflammation and see how important it is that local inflammation to destroy microbes and whatever we're talking about and have access to even the inside portions. So all those particles, they are what we call antigens. Antigen stands for antibody generator. So almost anything can be an antigen. So anything that is able to activate a lymphocyte, B, and form an antigen, an antibody, is an antigen. So these could be small proteins, could be bigger proteins, could be bigger portions of the bacteria or virus, could be carbohydrates, and so on and so forth. Okay, so when we say that the acquired immunity is driven by antigens, it means that it will produce something specific to that certain antigen. So let's try to work with this concept for a little bit more. So we have two types of lymphocytes that are involved in antigen-driven immunity, which are the B cells or B lymphocytes and the T cells and T lymphocytes. There is a very outdated definition, but some textbooks still include it, which is the humoral and the cellular immunity. Humor stands for fluid, and so we would normally include the B cells into humoral immunity in the sense that B cells produce something that is fluid, which are the antibodies. The T cells or T lymphocytes, on the other hand, don't produce that component that is fluid, and therefore we call that the cell-mediated immunity. The boundaries between these two kind of immunities are very changing, and so this separation may be somewhat outdated. But what's important to understand is that both of the B cells and T cells come from the lymphoid line in the bone marrow. But then the B cells mature in the bone marrow. So the maturation occurs still in the bone marrow, whereas the maturation for the lymphocytes the T lymphocytes, occur in the thymus. So the T actually stands for T thymus-dependent lymphocytes. So if we remove the thymus, we would not have T lymphocytes. Okay. This picture already tells a lot, but you can come back to it to review. So the T lymphocytes and the B lymphocytes react to specific antigens. So again, it's not that they form, we can create cells, in the sense we can create cells that are specific for a certain antigen. It's more than out of our genetic variability, we have a certain number of lymphocytes that will respond best to certain antigen. And so when we interact with that antigen, that lymphocyte will be stimulated, and then we will have memory cells and also activated cells. So they react specific. We don't form antibodies to become specific to an antigen. They are specific to begin with. So all these processes, I just wanted to interconnect, and this slide interconnects with the other component, with the component of the macrophages and all the other cells that perform phagocytosis, in the sense that by breaking down particles, these cells will allow antigen formation. So imagine that the antigen is a piece of a protein or a carbohydrate or lipid, whatever it is, inside of a microbe. Only when the microbe is broken, that substance can be released. So that's one role of all the cells that perform phagocytosis. But there are other interactions. For example, macrophages secrete interleukin-1, IL-1. IL-1 is a very important interleukin, for example, for mediation of fever. It's one of the mechanisms for fever, but it also stimulates, for example, lymphocytes. And we can also speak the other way around, how lymphocytes influence and activate macrophages and neutrophils. So, and we saw the receptors for IgE in the mast cells, in the basophils, and the eosinophils. So the system is much more dynamic. To add to this complexity, the system collaborates with the lymphocytes collaborate with the other cells, but they also collaborate with one another. The degree to which they collaborate is very variable, and it can be covered different aspects. But what is important to understand is that we need the T cells to activate the B cells. And that's what gave the name to the CD4 T cells to be T helper. Helper in the sense they would help the B lymphocytes. But in fact, we will see that the B cells can also help the T cells by presenting antigens to the T cells, by showing the antigens to the T cells. I wanted to just, again, it's the problem of teaching complex topics in a simple way is that we were never satisfied with the way we introduce certain topics, because we always feel that we are leaving things behind. But to give you a certain example here, HIV affects T helper cells, the CD4. And you see, if a person doesn't have CD4 cells, everything gets compromised. Many aspects of the immune system get compromised. So it's almost like an orchestra. All the pieces need to be working to have a full response. So you see here in this diagram how the T helper is connected to the B cell, and that activates the B cells. So let's now, once we get these ideas of collaboration out of the way, so out of the way in the sense we cover them, but we have to keep them in our mind when we speak about immunology. Let's go over each of those, the B cells and the T cells. So the B cells are when they never saw an antigen, we have what we call naive B cells. So these naive B cells will be out of genetic variability, specific for certain antigens. So let's imagine this is any lymphoid organ. So we see these three components here. We see a naive B cell, a naive T helper, CD4, and a dendritic cell. Once this tissue gets access to antigen, the B cell will be activated. It's what we call the antigen challenge. So once there is antigen being presented either to the dendritic cells or to other cells, they can present, let's put this sequentially because it makes more sense, but things are a little bit more dynamic. They present this antigen to the T cells and the T helper, the CD4, and the CD4 will be able to activate the B cells. Once the B cells are activated, they transformed into plasma cells. Plasma cells are the cells that produce antibodies. Okay, so things get a little bit more dynamic than that, because in this picture we show, for example, that a dendritic cell, which is a professional cell that presents antigens, is presenting the antigen to a T cell, a CD4, and the CD4 will stimulate the B cell. But note that the B cells can also help the T cells by presenting antigens to them. Once we have an activated B cell, the cell transforms itself in what's called a plasma cell. The plasma cell produces antibodies. So what are the antibodies? So the antibodies have two portions. A variable portion, which is the portion that varies and will be able to bind different antigens, and it has a portion that is constant, Fc. So it's almost like having a fork, and so the handle of the fork is the same, but or better, a key would be a better example. So there is a portion of the key that is the same, but the actual top part of the key is different from one another, varies depending on the key, on the key lock. So the antibodies are the same thing. The constant portion interacts with several aspects of the innate immune system. We talk about neutrophils, we talked about eosinophils, mast cells, and basophils, but we also will speak about the complement. The variable portion is the one that binds to the antigen. So each antibody has a specific antigen that they should bind best to. And we have 5 types of different antibodies. I want you to know a little bit about the following that I have in the different slide, which are the IgM, the IgG, the IgA, and the IgE. The D is not very clear what it does, so we can leave it aside. The IgM is the one that is mostly produced the first time the naive cell, the first time a B cell was activated. So that first time around when we have the plasma cells was the first antigen challenge. So the first time we interact with that antigen, we are producing plasma cells that produce predominantly IgM. IgM is very big. IgM is big enough to, and the size of it matters for 2 reasons. One is that it's a potent activator of the complement. And the second aspect has to do with the fact that because it's too big, it cannot cross the placenta, which is important to understand, for example, the Rh incompatibility and the ABO incompatibility between the mother and the fetus. We will mention that in a little bit by the end of today's lecture. IgG, on the other hand, is predominant the secondary response. In the secondary response, in other terms, the second time around, once we are seeing the antigen, not for the first time, but for a second or third, or so on and so forth. So IgM and IgG can be used to see if the person is immune to something or not. As we gain immunity, as we colloquially speak, we transition from IgM to IgG. IgG crosses the placenta, and this will be very important to provide the newborn with protection for as long as the IgG lasts, which is normally thought that would be around 6 months of the infant's life. IgE is important for allergies and the parasitic infections. And IgA is not a very strong immunoglobulin, but it shows up in secretions such as saliva and milk too. Okay, so let's just put this in perspective, but everything I said uh, you need to know. But let's put this in a form as a table. The only thing that matters there are the size. I have an idea that IgM is the biggest, the largest molecule. I have an idea that IgM activates the complement the best, and IgG crosses the placenta, and IgA is normally the one that we associate with mucosa. So, for example, in any secretion. Okay, and that's pretty much it. So how do antibodies help out? Well, the antibodies help out through these four mechanisms, and we have to just spend some time just thinking about their names. So the first is neutralization. So again, the idea that you can imprison the antigen. Imagine that the antigen is a toxin. So let's create this example that we are trying, there's a toxin that's produced by bacteria, and so the antibody neutralizes it. By binding it to it, neutralizes it. It can also perform agglutination and precipitation. Agglutination is when it's almost like tagging. The different binds to the bacteria. Let's keep the example of bacteria here, and then allows the bacteria to connect with one another, to clump together, to agglutinate. So you can see how this is almost like, you're trying to remove dust or remove any particles. So putting them all in the a big conglomerate of particles is helpful because you neutralize them simultaneously. You also make it easier to remove them. Precipitation is an equivalent component, but is with the antigen and the antibody. And lysis requires the activation of the complement, which we'll talk in a little bit. So to fully understand these concepts, we need to go over what the complement is to fully kind of get this, the lysis aspect. The rest is kind of clear. Neutralizing because it's interacting with the antigen in the way that antigen becomes ineffective, agglutinating, bringing cells together, and precipitation, making a cluster with the antigen and the antibodies. The lysis requires the complement. The complement resembles, to a certain extent, the clotting cascades, in the sense that there are different ways of activating the complement, but once a particle is activated, they will activate other particles. There are many ways of activating the complement. We talk about the alternative way, the classical pathway, and the lecithin pathway. The most important for us, because we can't have the capacity of covering everything, the most important for us to speak about is the classical pathway. The classical pathway is activated by an antigen antibody. So when the antibody binds to an antigen, it will activate the complement. And you have these particles. So you have C1 activating C3, C3 then forms C3b, C3b then is responsible for activation of other components until they form a complex. So there's several things that these particles may form, all these complement particles may form. And so I wanted to spend just the next 3 slides on this. So here it is what the complement serves. So one aspect is the membrane attack complex, which we'll have a slide just for it. But certain of these substances, all these C3 something, they have in a variable degree, they act as substances that can perform chemotaxis, so attract new cells, but also are inflammatory mediators, for example, for vasodilation or increased permeability. So we can put these two functions aside, but we are going to focus on the membrane attack complex and on the other one that's called opsonization. So let's start exactly with this one. So what's opsonization? Opsonization is tagging, it's creating a tag, really putting a tag on a microbe. How does it work? So the C3b, which is one of the most important opsonins, binds to the microbe. And now you have to remember that neutrophils, for example, have receptors for C3b. So that tag will allow the neutrophil to identify and to bind to the microbe that is marked for destruction. So that's called opsonization. Opsonization makes the process of phagocytosis more efficient. The other aspect is the membrane attack complex. Once we have the cascade complete, we have the following complex, which is C5b6789. This is a complex of all those proteins that they act essentially by creating a big pore onto the membrane. Well, what a wonderful way for you to review what the function of the membrane is, because if this complement does it to a bacteria, so again, all these mechanisms we've been talking about can be applied even in autoimmune disorders against our own cells, but we give the example of bacteria because it's a little bit easier for us. And so that would be destruction of the permeability, the selective permeability, and then the cell will die. Okay? So I would pause here and just go over the B cells and the complement, because now we are going to transition to the T cells, and yes, in the end we should put everything together, but it's best that you get the B cells correct and then we move to the T cells. So after you pause, you come here and we will be transitioning to T cells. So T cells are very complex. There are different kinds of T cells, and what it's important to understand is that they too depend on antigen. They mature in the thymus. Those are the two components. There are many types of T cells, but it's creating a summary like this is very oversimplified. But let's try it and see if we can keep this idea. So T helpers, we can allow the name of the cells to give us a little bit of the function of those cells. The T helpers were thought to be helping other cells. So those are the ones that are infected by HIV. Okay, so keep those two facts in your mind, and add the other one we spoke already, which is the fact that they are CD4 positive. Then there are two other types of cells we have not spoken about: the cytotoxic T cells and the regulatory T cells. The cytotoxic T cells are responsible for killing other cells. What cells need to be killed? Well, any cell that is infected or that has some abnormality. For example, viruses need to live inside of cells. So when a cell is infected by a virus, there are particles of the virus that will be presented, and they may tag that cell for destruction and is destroyed by cytotoxic T cells. But this may also happen, for example, in cancer cells. The regulatory T cells, sometimes called suppressor, have a more ambiguous role, and they are oftentimes thought that they modulate processes, which is a very simplified way to say it's very difficult to sum what they are doing, to sum up in a sentence what they are doing, so we'll call them regulatory. Okay, So the aspect here is that is different from the B cells is not only that the B cells produce antibodies and the T cells don't, is that the T cells need some other cell to present antigens. So they don't have the capacity to bind to antigens. So the antigen needs to be shown to a T cell for the T cell to be activated. And how are antigens presented? Antigens are presented on MHC proteins. And we'll go over just a little bit in a second what MHC is. We'll spell it out. But for now, let's just keep this on our minds, because I still want to talk about what kind of cells present antigens. Note that, at the end of this lecture, you'll know a lot of things, and the things you know at the end may help you understand some of the things that we've talked in the beginning, exactly because things are very dynamic when it comes to immunology. So let's talk about what cells can present antigens. We can speak about cells that present antigens as being antigen-presenting cells. And of all the cells, the most effective are the dendritic cells. There are dendritic cells everywhere, but when we talked about the dendritic cells in the lymph node, in the sense that they would come into the lymph node, they come with antigens, so they're very strong at presenting antigens. But again, many other cells do, such as macrophages and B lymphocytes. Now that we know which cells present antigens, let's work on these two concepts, and then we'll expand and we'll understand that we have many cells that present antigen. These ones are the antigen presenting cells, but they're called professionals. Those are called professional, so they are more effective. Let's just look at the molecules first, and all these becomes clearer. So there are two types of MHCs. MHC, first of all, stands for Major Histocompatibility Complex. So histocompatibility, so histo comes from tissues, compatible. The name kind of suggests exactly what it is that we can analyze the MHCs to see how people are compatible for transplants. So the compatibility between the donor and the receptor of transplants. We have two types. We have the class 1 and the class 2. Class 1 is presented by all nucleated cells. So red blood cells will not present, but any nucleated cell in the body has MHC class 1. The class 2 are presented by the APCs, what we call the professional APCs. So we're talking about the dendritic cells, we are talking about the macrophages and the B cells. I wanted to have an idea what it looks like. So we're talking about these components. So we have an MHC class 1, MHC class 2, and they have a little peptide where they bind antigen. So now let's work on these two components, okay? Granted that we already know MHC class 1 is antigens that are inside of the cell. MHC class 2 will be antigens that are outside of the cell. Let's just put this in perspective, and we need to now talk about the cytotoxic T cells. So cytotoxic T cells are CD8. And here for mnemonics, we can create this rule of 8. So CD8 cells bind to antigens that are presented on MHC class 1. 1 times 8 equals 8. So where does this antigen come? So the cell can be either infected, it can even be a cancer cell, or a cell that is for whatever reason change, such as changes that occur through aging. But the most classic example is the viral infection. So virus need cells. So the virus controls the cell to a certain extent, and there are viral proteins that are being produced. These proteins are not the cells, so are not our own bodies. So they will be presented in an MHC class 1, and they'll be identified by the cytotoxic T cells as foreign. And that will induce a response against this cell. That's why it's called cytotoxic. So they kill other cells. I want to give you just one more picture to expand on this. So it's all nucleated cells, and it can be anything. For example, a virus or a tumor antigen. So you see how it's being produced and how it's being presented on the cell. What about MHC class 2? MHC class 2 are presented by the APCs, the professional APCs, and so we are dealing essentially with something that comes from the outside. But I have a slide for that, so let's just put that idea there. But what matters here is to come back to the 8 rule. So the APC cells have MHC class 2, and they present antigens to T helpers. So here for these lectures is really a simplification, but you have to know everything so that I've told you. So go through the study guide, and you can pause here. But before that pause, I want to give you another image just to really emphasize that MHC class 2 present to CD4 T cells, and these antigens come from the extracellular component. So it could be microbes, for example. Okay, and then you can connect everything. You can just say, okay, there's the splinter here on the tip of the finger, and then the macrophage neutrophils will break down the microbes. Dendritic cells and antigens go into the lymphatics, and now you see the activation of T cells and the T helper cells, and then see how the helper cells activate the B cells. So the process gets much more beautiful in terms of knowledge. I wouldn't mean complicated in a bad way, but beautiful because it becomes more complete. And yet we are just learning really very simple ideas. So I would pause here to actually go see if you can explain the B cells and the T cells, and maybe go through the slides and see if you can now put things, expand that picture of the inflammatory processes. When you come back, I want to talk a little bit about a couple more of things. So MHC is important for self-recognition. And so another word for the MHC is called HLA, human leukocyte antigen. And so in great part, MHC marks self, non-self. So normally the immune system is, if everything goes well, it should respond to things that are not ours. So we don't want that immune system to be destroying our proteins, our carbohydrates, or our molecules. We want the immune system to destroy what is others. And MHC are unique to an individual. And therefore, they will also be important markers to help, for example, understand what would be compatible as far as an organ transplantation, because some are more important than others. The aspect that is very important to understand vaccination and so on and so forth has to do with the aspect of memory. So again, this is a matter, we can also look at this as like a military, an army. So imagine that, let's make this very like Simplified, imagine that the first time we interact with an antigen, we only have, out of variety, our genetic variability, we have only 10 cells, 10 B cells that actually were there, and they are able to recognize that antigen. We have 10 T helper cells that were able to recognize that antigen. So that first time around, we interact with that antigen, not only we produce more B cells and T cells that are going to be acting against that microbe, but we also produce memory. And these memory cells will stay dormant until the next time we interact again with that microbe. But the second time around, we will not have 10, we will have hundreds. The numbers are just really to illustrate the purpose. So once the lymphocytes have been challenged, they form memory cells, and these memory cells increase the army. So we can speak about a primary response and a secondary response. A primary response is dominated by IgM and is much weaker. But the secondary response is dominated by IgG. That we already know and we covered, but it's much more efficient because we'll have a much stronger response. So what is vaccination? So active vaccination includes exactly this, challenging our lymphocytes so that the next time we interact with the antigen, in the case of an infection, we are going to be much better prepared. So let's walk you through these pictures, and these pictures may have details that I did not mention. You can just not worry about them, but I want to show you these pictures so that they help you understand the overall process of memory. So here we have the naive cells, the exposition or the challenge to the antigen. B cells, T cells will be activated. See the role of the T helpers activating the B cells. The B cells becoming a plasma cell, and the plasma cell has IgM mostly. Simultaneously, you produce memory cells, which means that the second time around, we have more cells, and the response is going to be much stronger. So that graph shows you the amount of IgG that is produced. We produce much greater IgG in our secondary response. Okay, so pause here and go over this on your own and try to see if you understand these concepts. When you come back, I want to finish with, before we go into clinical correlations, I want to talk about innate lymphoid cells. So innate lymphoid cells are lymphoid cells that do not respond to antigens. The best of those are the natural killer cells. The natural killer cells do the same thing that, this is just an image to show you what these innate lymphoid cells are, but what I want to show you is about the natural killer cells. The natural killer cells kill other cells in the same sense that the cytotoxic kill other cells. But the cytotoxic cells respond to antigens. The natural killer cells are not driven by antigens. But both the cytotoxic T cells and the natural killer cells are killing our cells, whether they are infected with a virus or they are altered, such as in cancer. So these two cells may be used for immune-based treatment for cancer. So I'll just show you here. This is a complex diagram, but it doesn't matter for us, but just matters this little point that you have a tumor cell and the natural killer cell being activated, it will be able to kill, for example, a tumor cell. The last aspect before clinical correlations are the cytokines. So we didn't go over the cytokines, but just we talked only about the interleukin 1, which is important, for example, for fever. But so there are, the lists would fill up many more slides. There's different kinds of interleukins. There's interferons, which are also cytokines. There's something called TNF alpha, which you might hear about in the context of inflammation, and so on and so forth.